bodies must constantly regenerate and create new cells in order to grow and develop. But how do we get from a single cell that results from the fusion of a haploid egg and sperm, with haploid in this case meaning one set of a gene, to the multicellular diploid organisms that we are today, and diploid meaning two sets of each gene? In this video, we'll be exploring how genetic material is organized and passed down, how cell division works, and how both cell division and differentiation can lead to complex, specialized systems. The blueprint for passing down and coding a cell's genetic information is found within its DNA. An organism's DNA molecules consist of long strands composed of nucleotides, which are these little things connecting the two strands, and is located inside the nucleus of each cell. Did you know that the DNA in a single human cell, when stretched out, is about 6 feet long? I'm not even that tall. In order to package all of this genetic material efficiently, each strand of DNA is coiled around proteins to form compact structures known as chromosomes. And so there's really tightly compact and coiled. And I'm going to change this color. These are our chromosomes. So chromosomes come in homologous pairs. With one set inherited from an organism's female, and I'm going to use orange as female, the female set, and one set inherited from the male parent. And this one, we're going to use this teal color, male. Different sexually reproducing species have different numbers of chromosomes pairs in each cell. So, for example, humans have around 46 chromosomes that can be categorized into 23 from the female and 23 from the male. They essentially, each set of 23 will code for the similar kinds of genes, but we have a version from the female side and a version from the male side. However, not all animals will have 46 chromosomes. In fact, chickens, that does not look like a good chicken, but chickens actually have 78 chromosomes, which means they have 39 homologous pairs from the female, as well as 39 from the male. The chromosomes within each homologous pair have the same types of genes, like I said before. Some, for example, can code for hair color, but can have different alleles. And this is a new term, alleles, or variations of those genes, kind of like an allele in hair color that specifies brown or blonde hair color. Okay, time to put on our little construction hats to figure out how the genetic blueprints in chromosomes can be passed down from a parent cell to create two identical daughter cells. Imagine these are little cells. If we wanted to construct two houses that are exactly the same, I don't think guesstimating the instructions and measurements would be the best idea. Instead, we would need a variety of tools and specific steps in order to build the house in the same way. Similarly, the cell uses a specific process to ensure that the two daughter cells are genetically identical to the parent cell. This process is known as mitosis. Let's zoom in on our parent cell. For simplicity, let's assume that our parent cell has 3 chromosomes in its nucleus. N equals 3. When the cell is preparing to divide, each chromosome undergoes DNA replication to create identical copies that remain connected at the center as sister chromatids. So this is that's our new copy. They remain connected at the center, and so each side of this is going to be a sister chromatid. In the early phases of mitosis, the duplicated chromosomes condense to form this familiar X shape that we see under a microscope, right here. Each side of the X, like we said before, is a sister chromatid. As the cell prepares for division, the nuclear envelope begins to disintegrate surrounding the chromosomes. And the mitotic spindle, here I'll use it as yellow, begins to form on each side of the cell. Think of the mitotic spindle as a fibrous protein structure that can reach out and grab onto the chromosomes. And now we have all the materials and toolkits to start building our identical houses. As we move on to the middle phases of mitosis, the mitotic spindles will reach out and attach to the chromosomes that have lined up to the center of the cell. So our chromosomes are now both duplicated and lined up at the center of the cell. Going back to our yellow right here, each one is going to reach out and connect to the center. Now, proteins attached to each sister chromatid will allow them to walk along the mitotic spindle in opposite directions. So if we zoomed into one of these chromosomes, this half would be pulled by the mitotic spindle towards that side, and this half would be pulled towards that side. Evidently, each one of them would kind of come apart and being pulled to opposite sides of the cell. However, both sides of this tug of war will be winners because identical genetic material ends up at both sides of the elongated cell. So we drew our elongated cell. We would have 1, 2, 3, and 1, 2, 3. Both of our houses are now constructed and ready for the finishing touches. Imagine these are identical because my drawing skills are not that good. In the late phases of mitosis, the mitotic spindle begins to break down since it is no longer needed. A new nuclear envelope forms around each set of chromosomes as they begin to unravel back into less condensed forms. So now they're loosening up, unraveling, and as a result, this original parent cell is ready to split into 2 separate cells. So lastly, cytokinesis occurs, dividing the cytoplasm using a protein ring that pinches and squeezes the membrane into 2 separate daughter cells. And now we have one daughter cell here and one daughter cell here, both of which have identical genetic material as our initial starting cell. Our houses are now perfectly constructed prime real estate. Now going back to the question I asked in the beginning of the video, how we got from a single little cell to who we are today, we can point to mitosis as the process for how a single diploid cell with paired chromosomes resulting from the fusion of haploid gametes, remember that egg plus the sperm, can replicate to create an organism with many, many cells. But not only is mitosis important for organism growth, but it's also important for tissue renewal. If you've ever scratched the surface of your skin and seen white marks left behind, like little white marks on the surface of your skin, those are actually dead skin cells that have accumulated on the skin surface. Your body actually undergoes mitosis all the time to replace new skin cells and other types of cells that have died or worn out. Okay, so yes, I know what you're thinking. Because multicellular organisms grow by mitosis, this creates genetically identical cells, right? So, how do different kinds of cell types arise if they're all the same? The answer is that specialized cells only use a subset of their genes to make proteins. You have all this different DNA, right? But only certain parts of the genes will be used and carry out to make our beautiful proteins. So each cell type creates a unique protein combination that allows it to carry out its function. In other words, cells don't make proteins from all of their genes at once. That would be silly. There's no point in making things that aren't needed. That's like asking a fast food restaurant in a mall food court for a cute winter coat instead of the clothing store nearby. So both cell division and differentiation produce and maintain specialized cells, tissues, and organs that work together to meet the needs of the entire organism. Let's give ourselves a pat on the back for doing a good job of functioning efficiently every day. That's that's basically the minimum that we all need to do. And that's good enough. So, what are some of the key takeaways from today's video? Number one, a cell's genetic information is organized into chromosomes. Remember that kind of X-like structure underneath the microscope? Number two, mitosis distributes identical genetic material to two daughter cells. Imagine this as our two identical houses that have the same blueprint of DNA. Number three, mitosis occurs through a series of carefully organized steps. Remember the replication and the alignment in the middle and pulling with the mitotic spindle. Number four, mitosis allows for organism growth as well as renewal. And number five, cell division and differentiation can lead to organism complexity. Remember, they have different kinds of protein subset that can be transcribed from the same set of genes.